G. Blotting techniques. DNA, RNA, and proteins are separated by blotting techniques. These are described below. 1. Southern blotting techniques. A method developed by a molecular biologist E.M. Southern 1975 for analyzing the related genes in a DNA restriction fragment is called Southern blotting technique. Southern blots can easily provide a physical map of restriction sites within a gene located normally on a chromosome, and reveal the number of copies of the gene in the genome, and the degree of similarity of the gene when compared with the other complementary genes. See the diagram below. Figure 6.12. Procedure of Southern blotting technique. DNA isolated from a given sample is digested with one or many restriction. Figure 6.11. Consequently, DNA fragments of unequal length are produced. This preparation is passed through agarose gel electrophoresis, which results in separation of DNA molecules based on their size. DNA restriction fragments present in gel are denatured by alkali treatment. Gel is then put on the top of the buffer saturated filter paper. Upper surface of the gel is covered with nitrocellulose filter and overlaid with dry filter paper. The dry filter paper draws the buffer through the gel. Buffer contains single-stranded DNA. Nitrocellulose filter binds DNA fragments strongly when come in contact of it. After baking at 800 C, DNA fragments are permanently fixed to the nitrocellulose filter. Then the filter is placed in a solution containing RNA, a denatured DNA probe of known sequences. These are complementary in sequence to the blot-transferred DNA. The radiolabeled nucleic acid probe hybridizes the complementary DNA on nitrocellulose filter. The filter is thoroughly washed to remove the probe. The hybridized regions are detected autoradiographically by placing the nitrocellulose filter in contact with a photographic film. The images show the hybridized DNA molecules. Thus, the sequences of DNA are recognized following the sequences of probe. 2. Northern blotting technique. Southern blotting technique could not be applied directly to the blot transfer of mRNA separated by gel electrophoresis, because RNA was found not to bind with nitrocellulose filter. J. C. Alvine and co-workers in 1979 devised a technique in which RNA bands are blot transferred from the gel onto chemically reactive paper. An aminobenzyloxymethyl cellulose paper, prepared from Whatman filter paper number 540 after a series of uncomplicated reactions, is diazotized and rendered into the reactive paper and, therefore, becomes available for hybridization with radiolabeled DNA probes. The hybridized bands are found out by autoradiography. Thus, Alvine's method extends that of Southern's method, and for this reason it has been given the jargon term northern blotting. Like Southern, there is nothing northern or western. These blot transfers are reusable because of the firm covalent bonding of RNA to the reactive paper. The chemically reactive paper is equally effective in binding the denatured DNA as well. Small fragments of DNA can more effectively be transferred to the diazotized paper derivative than to nitrocellulose. These techniques were more being advanced, and more recently have been demonstrated that mRNA bands can also be blotted directly on nitrocellulose paper under appropriate condition. In this method, preparation of reactive paper is not required. The mRNA is isolated from the transformed cells and electrophoresed under such conditions that do not permit the development of secondary structures. The mRNA separated on the gel are onto nitrocellulose filter, which are then hybridized by single-stranded probe RNA or DNA. Thereafter, hybrids are treated with SL nuclease and RNase, which digests the single RNA DNA probe. It does not affect the double-stranded nucleic acid formed due to hybridization of RNA by the complementary sequences of nucleic acid probe. Structure of mRNA is revealed to the extent to which mRNA protects the nucleic acid probe. 3. Western blotting techniques, protein blotting or electroblotting technique. In 1979, H. Taubman and co-workers developed the Western blotting technique to find out the newly encoded protein by a transformed cell. Its working principle lies on antigen-antibody reaction, hence, it is an immune detection technique. In this method, radiolabeled nucleic acid probes are not used. This technique follows the following steps. 1. Extraction of protein from transformed cells. 2. Separation of protein by using SDS-paged sodium dodecyl sulfate polyacrylamide gel electrophoresis, where SDS acts as solvent for electrophoresis. 3. Transfer of electrophoresed gel in a buffer at low temperature 400 C for half an hour. 4. Blotting of proteins onto nitrocellulose filter paper. 5. Soaking of nitrocellulose filter, Whatman filter and Cos filter in transfer buffer. 6. Placing of Whatman filter paper on a cathode plate followed by stack of Cos filter, Whatman filter, electrophorus gel, nitrocellulose filter, Whatman filter paper, Cos filter stack, Whatman filter and anode plate. Figure 6.13. See the figure. Figure 6.13. Western blotting by using radiolabeled antibody and detecting the bands through autoradiography. 7. Putting the complete setup in transfer tank containing sufficient transfer buffer. 8. Application of an electric field 30 volts overnight for 5 hours to cause the migration of proteins from the gel to nitrocellulose filter and binding on its surface. The nitrocellulose filter has exact image of pattern of proteins as present in the gel. This type of blotting is called Western blotting. 9. Hybridization of proteins by using radiolabeled antibodies, 1-123 antibodies of known structure, isolated from the rabbit. 10. Washing of nitrocellulose filter with a wash solution tris buffered saline plus tween 20 to facilitate the removal of unhybridized antibodies. 11. Detection of hybridized sequences by autoradiography. The dots of diagram shows the presence of desired protein. H. Recovery of cells containing recombinant plasmid.